We are the Metro Turkey team, and I'm Şeniz Yüksel with my friends Hirin, Dorukcan, iGEM and Burak. We are going to present our project today. I'd like to start with some pictures. As we can see here are 4 pictures with actually the same problem. It's about pollution which affects the environmental and causes many health problems. Degredo, PETronum. So we like to say it all and get away from this problem, but it's not so easy. I like to start with some graphics about PET production. When we consider the material production in 1 year, we can see that 12.4% of them is plastic. And when we get a more specific look, nearly 265 million tons of plastic is produced each year. As the plastic production increases, of course, the recycling rates are increasing too. And after recycling, what becomes with that? Nearly 38% of them is landfill disposal, 35% becomes energy, and the rest is recycled. So what happens with that recycle? We thought, why don't we turn into something that a bacteria can use, for example, as an energy source, pyruvate. Our Metro iGEM teams and our instructors advisors came together and talked about it, what we can do to do this better. Of course, when we look at the PET chemical structure, we can see that there are polymerized monomer use of terephthalate molecules with repeating units, as we all know. And when we look at the PET projects made in previous years by the iGEM project, we can see 3 different teams. They always worked on PET projects, but they had always a byproduct. So we wanted to change it into something that the bacteria can easily use. Okay, now my friend is going to continue.
Thank you. And here we can see the map. Actually, it's a bit complicated, isn't it? Here. And here, have a look, detailed. And here the underlined pathway, yellow underline. And from the Catechol to the Pyruvate, actually, we are primarily focused on. And here is the structural analysis of them. We see the Catechol in the middle of the picture, actually, from Catechol to the Pyruvate and the PCA, and then Pyruvate, and it's joining to the TCA cycle. And here we see our project mainly to convert the PET into the Pyruvate pathway, and the PET is here. And what the previous iGEM teams do is that from the PET to the 2-hydroxymethionate semialdehyde, and from we are primarily focused on the from 2-hydroxymethionate to the Pyruvate here. And this is our basic circuit design. We have chosen the constitutive promoter to overexpress the enzyme activity, overexpress the enzymes, and between the constitutive promoter and the terminator, there is alignment of the enzyme, called parts. And here is our new part. We have obtained this new part from PCR production of E. coli BL21DE strain, and this is the new part that includes the 2-oxopan-4-enoate hydratase. The catalyzes the activity 2-oxopan-4-enoate into 4-hydroxy-2-oxopan-28. And here we see the gel limits of them. Right picture, we have seen the PCR product gel image, and the left picture, we have seen the part with the backbone. And our second new part is again obtained from E. coli BL21DE strain, and the enzyme is 4-hydroxy-2-oxyglutarate aldolase. But the organic compounds have many names, so it's also the 4-hydroxy-2-ketolarate aldolase, or another names as well. It catalyzes the activity 4-hydroxy-2-oxyglutarate into pyruvate, and it's written like glycolate, but actually it's also acetaldehyde. And what we have done for characterization is we have chosen the constitutive promoter. We have ligate the constitutive promoter with our first enzyme, the Catechol 2,3-deoxygenase here, and we have obtained the picture in the right. We have designed an experiment, and here 2 tubes. Two of them include the centrifuge specimen with its plasmid and enzymes. And white tube include, we've add to 0.1 molar catechol, and the color changes like that. And the left picture, we have seen the gel image of it. And here is our kill switch model. We have choose the arabinose promoter, and for the first, we have divided the kill switch model into two. First construct is work with while the arabinose is present, and antiholin and tet-LFA is expressed. Antiholin is for preventing the holin effect, and the tet-LFA is for suppressing the tetar promoter. You see the second circuit begin with the tet promoter, and when the arabinose is absent, the tet present cannot be suppressed anymore, and endolysin T4 endolysin and holin expressed, and those guys are for lysate the cell and cell death occur. This is the basic issue actually. When the arabinose is present, the cell is happy, but when absent, cell is dead. Here, don't just start.
Sorry for the troubleshooting. And when we're skipping to the modeling part, first I can show the schema of our modeling that our catechol diffuses to the cell and series of enzymes degrading to pyruvate, our main part. But what we are going to do in our experiment, we centrifuge all cells and burst them all to the environment, and the enzymes enzymes initiate with the catechol out of the cell environment and degrade catechol to the pyruvate. We are going to use MATLAB programs and biology, and you can see the catechol diffuses to the cell very high instantly, and due to the high instantly uptake, the pyruvate is going to increase linearly, and you can see the intermediate chemicals are going to increase a little bit because the pyruvate is increasing very high amount of them. And here are parameters of catechol degradation is taken by a series of enzyme databases like Brenda or XPASI, and we are taking these databases from NCBI. You can see all the parameters of, we are going to the catechol degradation. And our characterization used by 0.1 molar catechol is dissolves in our pure water, and we have divided two tubes in our, we have divided our part into two tubes, and one tube we add catechol and other tube we added non-catechol. After 4 minutes and 22 seconds later, catechol degrades the 2-hydroxymuconate semialdehyde, and it gives our green color as we expected, but in our second tube there is no color change. At last, when we are going to skip the kill switch model, first presence of the arabinose, the RSC promoter starts, and you can see that LOA and antiholin compounds is going to start increasing, and this inhibits the cell lysis, but you can see a little bit endolysin concentration a little bit high, but endolysin is start to initiate with the holin concentration, but you can see the holin concentration is not above 0, so it's not going to start endolyze and it's not going to lyse the cell, so our cell is live its functions. And second, in the absence of arabinose, our tetar promoter starts to initiate, and you can see holin and endolysin part is going to start in our cell, and these compounds start to degrade cell membrane and the peptidoglycan layer, so that the results gave us the lysis of the cell. You can see a little bit antiholin part is going to increase, but the amount of holin suppress the antiholin part, so antiholin did not communicate with the cell to not for suppressing the death of the cell. These are parameters of hill switch model that are taken by two iGEM teams, St. Andrews and iGEM Zurich teams. You can see all the parameters. And finally, from our experiment achievements, 2 new parts added to the library. You can see the parts name. And also we have done the composite and characterized a new part. And also 4 parts added by composite and to the library. You can see the all numbers. And here comes Helen. Yes?
Thank you. We also participated world's first interlab study on the synthetic biology field. There were 3 devices. The first device was already constructed by iGEM. We constructed the second and third device by doing digestion and ligation. Then we have transformed them. Then we had overnight culture, and by using these overnight cultures we made measurements by using flow cytometer. Here are the results. The first one is for control group, which only have LB and E. coli. The first line is for first device and second device here, third device here. And these are for the mean fluorescence intensity data. The first one is again for the LB and E. coli. The other ones are for the first, second and third devices. And that's all for the interlab study.
We also gave a lot of importance to policy and practice activities. We have divided our aspect into 2 main categories, the in-campus and the out-campus events. We have made a symbiosis survey for the first time. We have asked the students in our campus what they know about synthetic biology, how they, if they knew about iGEM competition, and we have also get the opportunity to get them know about our project about recycling, PET bottle recycling, and degrading them by a synthetic biology way. We have seen from the results, it was very surprising actually, for attending to iGEM since 2000. The main topics was related to microfluidics, synthetic biology, iGEM in METU in our school since 2007. Also, the other universities were interested in iGEM and wanted to participate to iGEM for the further years. They asked from us to advise them, help them, and we have helped them. Some of them maybe can start in the next year for collegiate team. We also had a PET bottle degradation game, which was a very enjoyable way for teaching people about our project. The rule was very simple: squeezing the PET bottles under 10 seconds, the squeezed most of them wins the game. Also, we have started the METU High School Ankara iGEM team, the high school team. We have helped them, we advised them, we gave lab education, and we are raising new iGEMers for the further collegiate teams. The out-campus events, here am I, and me and my team went to Eskişehir. We have met with the mayor of Eskişehir in Turkey. They were really interested in our project because it is a bit also a municipality issue. We have constructed a very big PET-like bottle, and a lot of people have thrown their PET bottles to that container, and it is a very good way to get attention and in creating an awareness in people's mind about recycling and about our project. We also met with the Atoms Turkey team. They are here, and we collaborated with them. We present our projects to each other and discussed. We have done 2 synthetic biology-based parties in this year. It was a very enjoyable, very attractive way to teach people about synthetic biology and also about our projects. The entire theme was related to synthetic biology. We drank some beverage from small laboratory tubes. That is the policy and practice part.
Thank you. And what we are doing for future aspect, actually here, we've thought that metabolizing the pyruvate increase in NAT and FAT enzymes that are carrier for the protons. And what we can do with them, actually, we can make them join into the MFC circuits, for example, to generate electricity. Imagine that degrading PET and generating electric, it's amazing, right?
We'd like to thank you for your attention. There are many people who helped us to get to this point. Thank you very much. And our sponsors, we'd like to thank them too. I'd like to also